Hello and welcome to TutorTube, where the Learning Center's lead tutors help you understand challenging course concepts with easy-to-understand videos. My name is Natalie Taylor, lead tutor for biology and chemistry. In today's video, we will explore the steps of transcription and translation. Let's get started. For today's learning objectives, we will learn the steps within the process of DNA transcription and translation, and we will be able to predict the ending polypeptide chains when giving a starting DNA template. What is the purpose of transcription and translation? Genes are encoded in the DNA sequence, but in order for those genes to be expressed, the DNA must be transcribed and translated. The steps of transcription and translation use the instructions from DNA to form proteins needed for organisms to function. Essentially, proteins are our genes being expressed. Transcription occurs in the nucleus of the cell. Usually, the entire length of DNA is not transcribed, only a specific portion of it. We call that specific length the transcription unit. A promoter sequence is a specific DNA sequence that is upstream, basically right before the transcription unit, that lets the enzymes involved in transcribing know where to start. Usually, the promoter box is called the TATA box because it is a sequence of DNA nucleotides TA, TA, AA, AA. Enzymes will identify this sequence and bind to the strand and begin transcribing from there. RNA polymerase is the enzyme that identifies the sequence and transcribes the DNA. It copies the DNA downstream of that TATA box. When it copies the DNA, it forms a copy of the DNA in mRNA, also known as messenger RNA. RNA is similar to DNA, but the structure of the sugar base varies slightly. Additionally, when transcribing the DNA, RNA does not have the nitrogenous base thymine. In its place, RNA uses uracil. Once the RNA polymerase reaches a termination sequence in the DNA, it will pop off the strand with the mRNA fully complete. The image on the screen is a representation of DNA being transcribed. RNA polymerase uses the 3' to 5' template strand to base the 5' to 3' mRNA strand on. But it can sometimes be easier to think that the mRNA strand sequence will be an exact copy of the 5' to 3' strand of DNA, with the exception of all the thymines being replaced with uracil. Transcription finishing touches. Before the mRNA can leave the nucleus to be translated, a few more steps have to happen. First, a 5' cap and a poly A tail are added to each end of the mRNA strand. The 5' cap is a specific guanine type cap, and the poly A tail is essentially just a long strand of adenines. The cap and the tail helps protect the mRNA from degradation. These two images show the 5' cap and the poly A tail being put onto the mRNA strand. Notice that the 5' cap goes on the 5' end of the strand and the poly A tail goes on the 3' end. mRNA have introns and exons, but only exons have sequences that encode for actual genes. Thus, the introns need to be removed. The spliceosome is what removes it. It essentially edits the mRNA strand by cutting out the introns. Remember, the introns are the intermediate segments between the important exons. Once the introns are removed, all the exons are combined and put together to make one complete strand again. The final mRNA strand is now ready to leave the nucleus. It's important to note that sometimes mRNA with introns and exons can be referred to as the pre-mRNA. The top image shows a eukaryotic and a bacterial gene. The red represents exons, and the orange represent introns. Bacterial genes, and other prokaryotic organisms, do not have any introns, so their mRNA have no introns in them that need to be cut out. However, in eukaryotic cells, DNA is a combination of introns and exons, which leads to introns having to be cut out of the mRNA copy. In the second image on the lower half of the screen, it shows the pre-mRNA strand being cut and spliced together. The yellow represents the introns and the blue represents the exons. The introns are removed and then the exons are combined all together. The mRNA strand can now be translated into an actual protein sequence. This occurs in the ribosomes of the rough endoplasmic reticulum. This means that the mRNA has to leave the nucleus to reach the rough ER. Ribosomes are a mixture of proteins and rRNA, another form of RNA. The rRNA acts as a binding site within the ribosomes. tRNA, aka transfer RNA, have a specific sequence of nitrogenous bases on one end and an amino acid on the other. The mRNA is fed through the ribosome where it reads the strand sequence in triplet codons. Basically, it is looking at the strand sequence in segments of 3. The tRNA with a complementary sequence to the mRNA triplet is then matched to the codon and attached. For example, an mRNA strand that had a triplet codon AUG would match with a tRNA sequence UAC. The A matches with U, the U matches with A, and the G matches with C. The complementary sequence is called the anticodon. The tRNA sequence that matches to the codon brings an amino acid to start a growing polypeptide chain. As the mRNA is translated down its strand, more and more amino acids are added to the chain, and they are being covalently bonded together. The ribosome has 3 binding units within it, the E, P, and A site. As the mRNA is fed through the ribosome, the tRNA comes in and attaches to the A site. The amino acid on that tRNA is then linked to the growing peptide chain at the P site. Then the tRNA exits through the E site. It's important to note that only 2 sites are ever filled at a time, either A and P or E and P. The image in the top corner of the screen shows the A, P, and E site on the ribosome. The mRNA binding site, basically where the mRNA strand is fed through, is also labeled in the image. The following slides show images of translation in action. In the first step, a new tRNA is bound to the A site. In step 2, the amino acid on the tRNA is then attached to the growing peptide chain. In step 3, the top subunit of the ribosome slides over and it moves the tRNA to site E and P. In step 4, the small subunit of the ribosome slides over and the tRNA in the E site is ejected out. Then the cycle repeats itself and step 1 begins again with the new tRNA strand binding into the A site. Which amino acids do the tRNA bring in translation? There are a total of 20 different amino acids, but with each spot in the codon having 4 base options, there are more than 20 possible codon combinations. So multiple codons can code for the same amino acid. Usually, when determining the corresponding amino acid for the template of mRNA, we match codons, not the anticodon, with specific amino acids. The following image on the screen shows a chart of all the possible codons and the amino acids they encode for. Notice that the codon AUG is highlighted red. This is because it is known as the start codon. Almost every protein sequence will begin with the codon AUG encoding for methionine. Also notice that UAA, UAG, and UGA are stop sequences, resulting in the end of the growing peptide chain. What do the amino acid chains do? The single polypeptide chain of amino acids is considered to be the primary structure of proteins. The chain will then bend, fold, and bond together to form secondary and tertiary structures, resulting in 3D structures of the protein. If multiple chains are folded together, it forms a quaternary structure. These proteins that the chain encodes for can be structural proteins or enzymes needed throughout the cell. To practice, let's look at this DNA template strand. 3' TAC, GCC, TAA, GAT, 5'. First, what's its corresponding coding strand? Essentially, what's its complementary DNA strand? Its coding strand would be 5' TAG, CGG, ATT, CTA 3'. Based off the coding strand, what would the mRNA sequence be? Its mRNA strand would be 5' AUG, CGG, AUU, CUA 3'. Remember, the mRNA strand will be a copy of the coding strand DNA with all the T's replaced with U. Now, let's practice translation on that mRNA strand. As a reminder, our mRNA strand is 5' AUG, CGG, AUU, CAU 3'. Based off this, what anticodon would match to each triplet codon? AUG would match to UAC. CGG would match to GCC. AUU would match to UAA, and CUA would match to GAU. Now, what amino acid string will the mRNA code for? The amino acid sequence will be methionine, arginine, isoleucine, and leucine. Remember, to find the amino acid sequence, we have to match the codon to the corresponding amino acid based on the charts shown earlier. To summarize, transcription occurs in the nucleus, where RNA polymerase makes a copy of the DNA strand into mRNA. Remember that RNA replaces all thiamines with uracil. Before leaving the nucleus, the mRNA must have the introns removed and a 5' cap and a poly A tail are placed on each end. Translation occurs in the ribosomes of the rough ER, where the ribosomes provide a binding site for the tRNA to read the mRNA and bind amino acid sequences together. The long chain of amino acids then fold on themselves to form a functioning protein. The images in this presentation were pulled from the OpenStax textbook, Crash Course, and the Essential Cell Biology textbook. Thank you for watching TutorTube. I hope you enjoyed this video. Please subscribe to our channel for more exciting videos. Check out the links in the description below for more information about the Learning Center and follow us on our social media. See you next time!